How's everything going?
Good. I was trying to find the food journal to turn in. It wasn't on Moodle.
It is on Moodle. It has been on Moodle for some time. I'll show you in a minute.
Okay.
Look on the week that it was assigned, not the week that it's due. So scroll up 2 weeks from today.
It says food journal 1. Then there's food blogging 2, but I don't know if that's the same thing.
Oh, there isn't a food journal 2, is there? Wait a minute. All right, you are correct. My bad. I thought it was food journal 1 was food journal 2. Give me just a sec, and I will add it. No, it's there. It's in today's date. My apologies. I thought I had added it when I put the blogging prompts up. All right. So since who was that? Was that Branson?
Blake.
A minute ago? Oh, it was Blake. Since you got us onto Moodle early on, let's continue with Moodle for just a minute. If you go to Moodle for this week, if you look at the 23rd September 2000 materials and open up that PowerPoint, you'll see that that PowerPoint has been updated. I've gotten rid of all of the genetic engineering notations or genetic modification notations and replaced them with genetic engineering instead. And the other thing that you'll see is there are some minor changes to some of the slides, primarily in that I have added a lot of annotations to some of the things that we're going to be talking about today. So if you open it up in PowerPoint and look at the notes section, you're going to see notes on those slides where there were not notes before. So if you saved the old PowerPoint to your computer, you can just delete that and upload the newer version of the same PowerPoint. I've put Food Journal 3 assignment up there just as a place to submit it for those of you submitting things electronically. Mary, I have your food journal was outside my door when I got in this morning. So there it is. So either bring it by my office if you're handing it in hard copy, or submit it to Food Journal 2, which is now at the very bottom of this. So Blake mentioned that there's no place to submit Food Journal 2. Well, there is now. So go ahead and submit that. There are new blogging instructions for September 25th through October 9th. So 2 opportunities to blog on point 1 and 2 opportunities to blog on point 2. Since we're talking about genetic engineering, I would like you to reflect on genetic engineering as it appears in your food supply, and considering how much corn and soybeans is in our food supply. It'll be interesting to kind of see what you make of examining your diet from the standpoint of what ingredients are actually in my diet, and what of those ingredients have probably been altered through genetic engineering. And then the other thing is just a kind of more fun thing to do than think about genetic engineering is we're now in fall. Fall turned at the beginning of this past week. Normally I make note of it at the beginning of the week, but since we were having tests, I didn't get that opportunity. But we turned from summer into fall formally on the 21st, 22nd of September. And now it's pumpkin spice everything. So strangely enough, in our consumer culture, some time ago, Halloween stuff started coming up. And I was just in Lowe's over the weekend, and I see that they have shoved the Halloween stuff off to the side, and Christmas stuff is now entering the home improvement store. And so we change seasons, it seems like, earlier and earlier every year. But now it's pumpkin spice everything. You can get your pumpkin spice latte at Starbucks, et cetera, et cetera. So this represents kind of food faddism. And so I'm just curious as to what fads. Oh, Winston, you can't hear. Okay, let's see. Can everybody else hear me? And Winston, you still can't? I'm gonna guess that's yes. All right, let me type something to Winston real quick. Let's see. Let me go to the participant screen, see if I can do something with Winston.
Can you guys hear me?
Yeah, we can hear you.
Yes. I can't hear you guys.
Got it. Give me a minute to see if we can't get Winston hearing us. I can't type for some reason. Winston's going to try and log on from his iPad because it's not working from his computer. Anyway, what was I saying? Oh, I was talking about food fads. So we get all kinds of food fads coming through our food supply periodically. I think at the beginning of the semester I mentioned that because I don't really eat out a ton, I was reading food journals of my students one semester, and all of a sudden all of my students were eating at Subway for some reason. I'm like, okay, what is the deal with Subway? And they're like, oh, $5 footlongs. I'm like, what? So they're like, $5 footlongs at Subway. I'm like, I have no idea what's that about. And they were basically like, well, you can get a footlong sub at Subway for $5. I'm like, oh, okay. You're poor college students. You're getting cheap food. I get it now. But at the time I didn't have a TV. I listened to NPR all the time. Subway does not advertise on NPR. So I was just culturally kind of out of the loop. Hey, Winston, can you hear us now?
Yes, I can hear now. I'm good.
All right, awesome, good, great. So anyway, are there any food fads that you have kind of incorporated into sort of your normal diet? In the book, Omnivore's Dilemma, Poland talks about his kids eating Go-Gurt, and the fact that his grandmother, if you gave her Go-Gurt, she wouldn't even know what to do with it. And so Go-Gurt, which is a yogurt that is in a little squeezy tube, and you rip the top off this tube and your kid squirts it into his or her mouth and they eat it. And if you handed it to my grandmother, she would struggle with knowing what to do with Go-Gurt. So 2 of the prompts are about genetic engineering, and then 2 of the prompts are about food fads and any food fads that you may have picked up as a regular part of your diet. So those are food blogging things. In addition, on this week's materials, there is a link to a CRISPR-Cas9 video that we're actually going to watch in class today. But it's also there so that you can go back and review it. I'm going to go through that material in slides, but it's sometimes more informative to actually watch a video that has really nicely done animations by the person who actually developed CRISPR-Cas9 as a technology. And then there's an interesting story that has appeared over about the last year and a half. And it's been covered in other places, but I particularly like the way that NPR has covered the story, simply because it gives it kind of a more personal note to it. There is a woman in Mississippi who has been CRISPR'd. She has sickle cell anemia, which is a genetic condition that affects primarily African Americans. It's a single nucleotide polymorphism in the hemoglobin gene in humans, and she has basically had her DNA altered to essentially repair that mutation. And they did a series, they've done a couple of interviews with her and her doctors. One appeared, I think, back in December of 2019, where she was about 3 months into the treatment. And then they did another one just this past summer that was kind of a follow-up. And so CRISPR technology, we're going to talk about today, how it works. But it has potential to do a lot of really cool things. And it also has the potential to do some kind of sort of scary things. And people have difficulty separating, promising from scary sometimes. And so hopefully the radio interviews will shed some light on that. And you don't have any idea what CRISPR-Cas9 is yet, so knowing those things really doesn't me telling you about these things doesn't really resonate with you yet. So what we're going to do is we're going to go through some material today in class. I'll show you the CRISPR-Cas9 video, and then as homework for Monday, I want you to go and listen to the NPR interviews over the weekend, so that on Monday we can come back and we can talk about this woman in Mississippi. So, questions? All right, then let me share my screen with you so that we can proceed. So we had just gone through kind of the history of genetic engineering with reference to genetic engineering in our food supply. And I was just about to start going into the early technologies that we used to do this when we ran out of time last time. But the last thing that I talked about is kind of how prevalent genetically engineered foods have become in our food supply. So when you look at an ingredient, do I have any food in my office? I should have thought about this better, more in advance. Ah, yes. Going through my trash can from the week, I have, well, okay, all right, I can't do this. Unfortunately, the only food that I have in my office is tea bags. And so on my chamomile tea bag, which I occasionally make myself tea because my throat gets sore as I talk all day, chamomile tea. The federal government requires that food be labeled. And so on the back of the chamomile tea, you can see that there's an ingredient list. And unfortunately, this ingredient list just lists chamomile. And that's not really very useful to us because I was hoping to have a food label here on the fly that had something more interesting than just the single ingredient in it. But any packaged food that you get from the store, if you flip it over on the back or on the bottom, you will see a list of ingredients. And some of those ingredients will look normal, you know, sugar, water, et cetera, et cetera. But then a lot of those ingredients will just be this alphabet soup of chemical names that food scientists have put into your product. Some of the times those things are preservatives. Some of those times those things are binding agents. In Michael Pollan's book, I forget where it is exactly, he talks about a chicken nugget and how a chicken nugget is primarily made up of corn. And he maintains that this is the case because we feed the chicken corn. The corn is then rolled in cornmeal. The cornmeal is held on to the nuggets by food binding agents that are developed from corn. The pieces of chicken in the chicken nugget are held together by binding agents that are made up of corn, et cetera, et cetera. And so when we grow corn, we then send that corn off to a processing plant in many cases. And in some cases, it's converted into, you know, sweet corn that you eat, you know, corn out of a can or corn on the cob. But in many cases, the corn that you eat is commodity corn. And that's the bulk of what you see when you drive down the highway. It's this corn that is left out in the field until it's dry. It's harvested dry and it's stored dry, and it can be crushed up and made into a wide variety of substances. Everything from the glue that holds the paper backing on drywall to the ceramics in the ceramic components of spark plugs to the binding agents in food. A lot of it also goes into animal feed. So the feed that we feed cattle in concentrated animal feeding operations and things like this, all of that stuff is what we call commodity corn, this dried corn that is harvested dried and stored dried. And so when you look at any of your packaged food, a lot of the chemical compounds that you see that have complicated names in your ingredient list, those are all compounds that are ultimately derived from food scientists and chemists developing products and compounds from corn. And so it's all corn based. And a lot of the protein that we get in our protein containing processed foods are actually from soybeans. And when you look at corn and soybeans, you see that around 90 or over 90% of the soybeans that are grown in the United States have been altered to have herbicide tolerance. And a good chunk of the corn, about 80% of the corn is BT corn, which once again has Bacillus thuringiensis, and about 90% of the corn has herbicide resistance engineered into it. And so given that so much of our diet is based in corn and soybeans, you can guess that probably a lot of the corn and soybeans that you are consuming has been genetically engineered, even though it might not be the raw corn kernels that you are using, but certainly the compounds that came from those corn kernels were genetically modified. Now, the chemical compounds don't have any genes in them. And so even though the kernels themselves may be genetically engineered, the chemical compounds are actually a series of steps away from that. So it's a lot to think about. It's a lot to process to think about. Okay, we altered this gene to make it herbicide resistant. What has that done to the plant in terms of what does that mean for the farmer in terms of their ability to grow a crop that doesn't have to compete with weeds? What does it mean from the standpoint of then spraying herbicides done on all of those fields? And what does it mean in terms of the actual chemical compounds, glucose, dextrose, anything else that is derived from that corn? Does that herbicide resistance really have anything to do with any of those things? So it's a kind of complicated critical thinking task that is before you in your food blogging this week. My hope is that it will also prompt you to then go and start reading the GMO report by the National Academy of Science, because I think the things that you read in there will help you contextualize this particular blogging task. So in the assignment, it asks you to talk about genetic engineering twice. My advice would be to do that once one week and once the second week, and it asks you to talk about food fads twice. So I would once again do that once one week and then again the second week, just to give yourself some time to wrap your head around the genetic engineering part of the assignment. Okay, so a lot of genetically engineered crops out there in terms of the bulk of what goes into our diet. So how do scientists accomplish this? So scientists are very good at studying organisms, and when they learn things about organisms that are useful to them, they're very good at taking these things that they learn and adapting them to be useful. And so what they did early on in the development of genetic engineering is they used the cellular mechanisms of other organisms to do work for them that they wanted to do. And so this is a bacterium, Agrobacterium tumefaciens. It's a type of bacterium that causes tumors in plant cells. And one of the ways that it does this is it does this by hijacking the genome of the plant that it infects. So in a bacterium, you have the main bacterial genome, the main bacterial chromosome. Bacterial genomes are organized around a single large chromosome for a bacterium, but then they also have smaller chromosomal elements, these things called plasmids. And plasmids are used by bacteria to move their genetic material from one bacterium to another in many cases, but they can also use it to move their genome or portions of their genome from the bacterium to a eukaryote, in this case, a nucleus-containing organism like a plant cell. And that's exactly what Agrobacterium tumefaciens does. When it infects a plant cell, it basically ejects this plasmid into the plant cell, and then that plasmid incorporates its DNA into the DNA of the plant cell. And so when scientists learn this about Agrobacterium, they were also like, well, can we use this mechanism that this bacterium has developed to get genes that we want in the genome of a plant into a plant cell? And the way they do this is basically using a thing called a restriction enzyme. So if you look down in the notes in this slide, you will see that I talk about restriction enzymes. Restriction enzymes are enzymes that cut DNA at a particular site. And the recognition site for restriction enzymes is just a sequence of nucleotides, you know, A's and T's and C's and G's. And they're specific, you know, a particular restriction enzyme only recognizes a particular sequence of nucleotides, nitrogenous bases, and will only cut DNA at that point. So what you do is you pick a restriction enzyme that will cut DNA at a particular place, and you want to cut DNA, you want to use restriction enzymes that will cut DNA such that it leaves free ends of DNA open. So you find a gene of interest. So let's say that you have a gene that makes a plant resistant to herbicide. Well, whatever genome that gene resides in, you want to pick a restriction enzyme that will cut to both sides of that gene, so that you have a segment of DNA that has the gene in the middle and then has a free end of DNA on one side and a free end of DNA on the other side. Then what you do is you cut the plasmid from the bacterium with the same enzyme. And so what you end up with is if you're using the restriction enzyme EcoRI, that cuts where there's an adenine thymine juncture here, and what it leaves you with is a free end with 4 nucleotides here and a free end with 4 nucleotides here. If you have then cut this gene out of another organism's genome with the same restriction enzymes, it leaves the same free ends open. And then this adenine, thymine can pair up with this thymine, adenine using the same rules of complementary base pairing that we studied back when we talked about DNA replication. This thymine, adenine pairs up with this adenine, thymine using base pairing. Therefore, you can insert that foreign DNA into the plasmid of the bacterium. You see that there's still this little gap there, so it's not completed, but there's an enzyme called DNA ligase that actually will form the bonds between that last adenine and whatever the next nucleotide is in the sequence, so that now you have a complete plasmid that has been healed up. And it's called a chimera. This is what we refer to as chimeric DNA. Do you guys remember from mythology what a chimera is? Anybody know what a chimera is? You might have learned about these things when you were a kid. Go look up what a chimera is. You guys all have computers in front of you. Also, if you've read Harry Potter or seen the Harry Potter films, they have chimeras in Harry Potter.
Is it something with like a lion's head that like breathes fire in a goat's body?
Okay, so a goat's body with a lion's head on top of it. You guys ever seen the statue of the Sphinx at Giza? That is also a chimera, a lion's body with a human head on it. So a chimera is just any organism, any mythical creature that is made up of the parts of 2 or more mythical creatures. So the Pegasus would be a chimera because it's a horse in all ways except that it has bird's wings mounted on its back and or shoulders. And so chimera refers to any of these mythical creatures that are made up of the parts of 2 or more actual existing creatures. And so chimeric DNA is DNA that is made up of DNA segments that are from 2 separate organisms. So once you've created this chimeric plasmid DNA, it has the gene that you're interested in getting into a plant, and it's connected to the plasmid, which the bacterium is capable of getting inside of a plant. Well, then what you will do is you will expose the plant to the bacterium that contains the engineered plasmid. That DNA then gets inserted into the genome of the plant cell. And then as that plant grows up, that plant will now have that gene in all of the cells of the plant, because through somatic growth, we start off with a single ovule that has been fertilized through pollination, and that single cell is going to then grow into all of the plant cells and tissues that make up the final plant. And so in this way, we can take a bacterium, alter the plasma to contain a gene that we want in there. We now have an altered plasmid. That gene then gets inserted into the genome of the plant, and then as a result, we now have a transgenic plant that contains that gene. Any seeds that are produced by this transgenic plant will also have that gene in those seeds. And so you can create a line of transgenic plant as long as you insert it into the plant body early enough that it affects all of the cells of the plant, including the cells that are going to become flowers and become ovules and become pollen. And so in this way, you can create a line of plants that has a particular gene of interest. The most common genes of interest that we have put into plants are genes that make you herbicide resistant, genes that make you produce your own bacterial toxins that negatively affect the insects that might feed on you, and so it produces its own insecticide. In the case of many of these plants, we've introduced genes into them that make them more drought tolerant, so that they can be grown in drier, hotter areas than they would normally be able to be grown in. And then in the case of rice, which is a staple in the old world, in the Asian portions of the world, rice is high in carbohydrates, but unfortunately it's low in protein. So some varieties of rice have been engineered such that they produce more protein. So in addition to getting a good supply of carbohydrates, you get a higher supply of protein when you consume rice. And so it's improved the diets of people in certain parts of the world because of improved protein content in their diet, if their diet is primarily based on rice. And so there are all kinds of genes that have been genetically engineered into organisms. Questions about this process? If you go back earlier in this slide presentation, you can see some examples of the different kinds of crops and what kinds of genetic engineering has been done on them. So just to remind you that that information is in here. Okay, we're all clear on process. Important for you to remember things from the first test, like complementary base pairing, which we talked about when we talked about DNA and we talked about mutations and things like this. That is important in genetic engineering. If we didn't have that complementary base pairing, where adenines naturally bond with thymines and cytosines naturally bond with guanines, this whole process of getting genes out of one organism and into another organism would be more difficult, because we would have to do more of the work rather than just chemistry doing a lot of the work for us. And so the information that we've seen in the past, don't forget that information. That information is still important now. It's going to become especially important here in a minute, because when we start talking about CRISPR, CRISPR operates in a way that is heavily dependent upon complementary base pairing. So that's why I wanted to call attention to that once again here. Questions? All right, so CRISPR-Cas9. CRISPR-Cas9 is a relatively new method. And what scientists did was they discovered that bacteria have ways of preventing themselves from getting infected by viruses. So there are a lot of viruses out there that just infect bacteria, and these viruses do the same thing that bacteria do to plant cells, viruses do to bacteria cells. They inject their DNA into the bacteria, that DNA gets incorporated into the genome of the bacterium, and then the bacterium becomes a factory for creating more versions of the virus. As it turns out, this is actually how viruses like AIDS and coronavirus work also. They inject their DNA into your cells, and then once it is incorporated into your genome, your cells become factories for creating more copies of the virus. And so in this way, viruses are fairly devious and clever about how they reproduce themselves. Well, what bacteria have done, because they tend to get infected by viruses, is they've created a way of disabling viral DNA. And so what they do is they create a guide RNA. This guide RNA has a sequence of nucleotides, a sequence of nitrogenous bases in it that matches part of the sequence of viral DNA. This enables this guide RNA to then complementary base pair with the DNA of the virus. So in this way, the RNA molecule that is created by the bacterium targets the DNA sequence of a specific virus. Then it uses that DNA sequence to combine that DNA molecule with a Cas9 protein. So this Cas9 protein is basically a general purpose protein that cuts DNA. So in the same way that restriction enzymes are proteins that cut DNA, Cas9 is another type of protein that cuts DNA. And in this case, it cuts DNA rather than usually with sticky ends. So we talked about restriction enzymes cut DNA such that there's an end here and an end here that allows for complementary base pairing. Cas9 tends to not, so these things are sometimes referred to in the literature as sticky ends. And they're called sticky ends because they're free-floating ends that want to attach to other complementary nucleotides, complementary nitrogenous bases. And so they tend to stick to other pieces of DNA that have a similar sequence, sticking off the end. So Cas9, though, tends to cut things, and this particular cartoon shows it as a kind of a Swiss Army knife with a little scissor attachment. Cas9 tends to cut DNA without making sticky ends. And so this protein is capable of cutting both strands of DNA separately. It can simply cut the DNA, and when it cuts the DNA, it inactivates the gene, or it can cut the DNA to open that DNA up to make it available for the insertion of new DNA sequences. And this is why the creators of this particular cartoon illustrate this as a Swiss Army knife. It is kind of a Swiss Army knife of cutting. It can cut DNA so that that DNA can have a variety of things done to it. Initially, people just used CRISPR-Cas9 to turn genes off by cutting the gene up so that it's no longer functional because it isn't in its full length anymore. But CRISPR-Cas9 can also be used to insert new segments of DNA into the target DNA at the place where the cut occurs. And when this does this, it can have a variety of different effects. The gene can either be turned off if it's just simply cut, but it can also be upregulated, meaning that the cell will produce more copies of the gene than it normally would, or it can be downregulated depending on what the action of the insert is that you put into this. And so there are a number of ways in which you can modify gene action using CRISPR-Cas9. And so CRISPR-Cas9 has been used a lot in experimental organisms. So one organism that is used a lot in genetic labs, as a matter of fact, Dr. Reynolds here in the biology department uses nematode worms, and you can use CRISPR-Cas9 to alter the phenotype of nematode worms. It's been used to alter the genotype and phenotype of fruit flies. And then I think this other one here is a zebrafish that has been modified using CRISPR-Cas9. And so this is rice being grown here on culture in a petri dish. And you can see that they converted normal rice into albino dwarf rice, once again, just by altering the genes in rice due to CRISPR. And so it's a powerful tool because you can basically take any gene from anything and put it anywhere in the genome of an organism. One of the advantages of CRISPR is the high degree of specificity that occurs with CRISPR. The challenge with using a bacterium like this is this restriction enzyme that you use to cut this DNA out of one organism's genome and put it into the plasmid of a bacterium. That enzyme cuts it not only right where you want it to cut, but it cuts it in all sorts of other places as well. And it cuts it in all sorts of places here and it cuts it in all sorts of places in the plasmid. And it also then will insert into a variety of different places in the genome of the organism that you're putting that gene into. And so it's much less specific in terms of where it gets incorporated into the genome. A lot of times, this new plasmid may get inserted into a genome such that it inactivates a gene that already exists that is really important for that organism developing properly. And as a result, that particular transgenic modification doesn't take hold because it ends up having lots of negative effects on the organism that has been engineered. So this ends up being quite a lot of trial and error by the time you finally get it right. What CRISPR-Cas9 allows us to do is it allows us to be very specific. We can design the guide RNA so that it does whatever it's going to do at a specific place inside the genome of the organism that we're targeting with CRISPR. This is the advantage of CRISPR-Cas9 over other ways. And when you listen to the article that I've posted about sickle cell anemia, is that in that case, you're targeting not just a specific part of the hemoglobin gene. So hemoglobin is special in that hemoglobin is actually made up of 4 different proteins. So four proteins that are all wrapped around one another. And sickle cell anemia is due to a single nucleotide polymorphism, meaning that there's one nucleotide that is different in all of those genes of all of those 4 hemoglobin molecules. There is one single nucleotide polymorphism that is responsible for causing sickle cell anemia. And so in order to do something about that, you have to be very specific in terms of targeting exactly where you want to change something in the genes that are responsible for forming hemoglobin. And so one of the advantages of CRISPR is this high degree of specificity. All right, questions about this so far? Okay, I'm going to try playing a video. Sometimes I have trouble playing videos because the sound doesn't want to come through, but this is basically the video that is linked on the website. So let me know relatively quickly here. Can you guys see YouTube? Okay, let me know relatively quickly here if you can hear the audio coming from this. Can you hear?
I can't.
No.
Cannot hear. Okay, all right. Hold on. Let me see if I can get it to work. Let's see. All right, let's try this. Can you see the McGovern Institute? Okay, now try. Do you have audio now? No. No. All right. Okay. All right. Let's see. Still nothing audio? No. All right. Okay, let's do this. I'm going to give you, this is a 4-minute video. I'll give you about 6 minutes. Go to Moodle, access, well, no, nah, never mind. Because I want to talk over this in some ways. Yeah. Let's see. Sometimes this works and sometimes it doesn't, and I haven't figured out what the secret is to getting it to work. I just kind of stumble into it. Okay, let me see if I can fix this. Is it still sharing the YouTube at all? No, okay, all right. There it is. Okay, let's try this again then. Okay. Okay. All right, can you see the YouTube? Okay, let's try this again. Can you guys hear it?
No.
I can't even hear it now. All right. So, all right, I'll talk it through this. I'll talk us through this. So, let me. No, I can't even hear.
Could just watch it on our own though.
Yeah, but I want to call attention to a couple of things about it. So. Okay. And then I'll probably just cut you loose and let you watch it on your own. And for some reason, it's now not letting me do anything. Can you guys still see the video? You can still see it, okay. All right, so here at the beginning, it's showing you, and this is calling attention to the complementary base pairing. So these different links, things sticking out, this is the sugar phosphate backbone of DNA, and these are the nitrogenous bases, and the nitrogenous bases will complementary base pair, adenine base pairing with thymine, cytosine base pairing with guanine. They use some odd language. They refer to it as the letters. They're represented by letters, but remember that they're nitrogenous bases. And so they'll talk about these things, complementary base pairing. They're essentially talking about the basic structure of DNA. So this is a good review for you. Just remember to translate it into the kind of language that we used in class. We're going to talk a lot about the uses of CRISPR, et cetera, and how it's going to be oh so great for humanity. And they talk a little bit about DNA sequencing and how that has made this possible and how we've identified lots of different genes in the human genome that are responsible for all sorts of maladies. Now they're going to be talking about DNA, and they're going to be talking about, this is the Cas9 molecule. And so this is DNA in a bacterium that comes from a virus. So here you see a virus, and it's going to inject its DNA into the bacterium, and then the bacterium is going to latch onto that DNA because it has created the RNA guide that then binds with the complementary sequence of DNA from the virus. That complementary sequence then allows the cell to hold the viral DNA rather in place, and allows the Cas9 protein to then interact with it at a specific part of the sequence. Once it's done that, the enzyme itself is responsible for cutting the DNA, and then what that does is it essentially inactivates the DNA of the virus so that it can't function any longer. And then they're going to go through and they're going to talk to you about the different applications of CRISPR. So they're going to talk about how then we can use that system from bacteria in other organisms to basically alter the DNA of those other organisms. And in some cases, once again, they use it to deactivate genes. In some cases, they'll actually use it to insert new genes into that genome. So rather than belabor that point, I'll just let you view the video on your own. And unfortunately, I cannot. There's. So, okay. So I've stopped sharing, correct? All right. So 3 things for Monday. 4 things for Monday. 1, view this video where you can actually hear the commentators. This is actually a video that was produced by the people who invented the process. So the actual person who developed this this video, that developed the technique, was actually consultant on this video, and so it's pretty factually correct. So watch this video. Listen to the 2 NPR clips about CRISPR and sickle cell anemia. And if you haven't started reading at least chapter 3 of the National Academy of Science Genetically Modified Organism report, you should start doing that because it revisits a lot of what I've talked about the last 2 class periods. And then we'll pick this up again on Monday. And as I promised at the beginning of the semester, when we get to the end of class and we've covered everything that was scheduled for that day and we happen to be early, I'll ask you if you have any questions. Do you have any questions?
Yeah, test. When should we be looking out for test grades?
I might have them done by Monday, but I might not get them done until middle of next week, perhaps. But I'm working on them.
Okay.
Other questions? This is a lot of heavy-duty science here. Are you guys following okay? I can't see most of your faces. So, and Mary just looks uncomfortable, like, I can't believe he's asking me if I understand this stuff because I don't really yet. Okay, all right. Watch the video, write down questions that you have about this. This is actually fairly complicated, fairly new science. And so I don't want us to just proceed as if you understand. I want you to understand before we then proceed. But the only way that I can know that you don't understand is if you tell me that you don't understand and we get to the basis of what's the lack of understanding. So I actually need you guys to help me with this process. All right, questions? Okay, then I expect to see some questions on Monday. And until then, get watching videos and listening to radio programs.